was my former PI for my second postdoc, and then I had a great time in his lab. He's keen to my heart. That's why I invited him to today to talk about Alzheimer's, which I think is a very important disease. And then it's like most of what I've been doing my entire life. So like it's more than 15 years of research on Alzheimer's, and then for Professor Klein, a little bit more than my time. So like just for those that are not familiar with him, Professor William Klein, as I said, is a professor at the neurobiology and neurology department at Northwestern University. He is an accomplished scientist. He has many publications, many awards, TED Talks. He's a very important figure for the Alzheimer's field, because he was the first one that introduced the concept of soluble amyloid beta oligomers as the main toxins for Alzheimer's. So like the contributions for his lab from his lab were like really a stepstone for the field that came like 10, 20 years after that. So like he has a lot to share with us today, and then I'm not gonna talk too much about him, because you will see through the data that he will show that he's been doing amazing stuff, and we are delighted of having you, Professor William Klein, which we also know as Bill for for us. We are happy to have you here. I'll leave the floor for you, and we are, as I said, very happy to have you here.
Mayra, thank you so much. It's great to see you, even through a lens here. I wish we were getting together in Brazil. I've been to so many wonderful meetings in Brazil next year, okay?
Yes.
All right. So thank you so much for inviting and for introducing me. I'm going to talk about the A beta oligomers that Mayra just mentioned, and then I'm going to look at them through a different lens towards the end of my talk. So let's start with the problem of Alzheimer's disease, which is such a serious and difficult one to contend with. There's over 25 million people around the world who have Alzheimer's, and in the United States alone, the cost in 2050 is estimated to be over $1 trillion. That's a whole lot of zeros there. It's a progressive deterioration, and that you can see from these self-portraits of William Udermollens as he was diagnosed with Alzheimer's disease and continued to paint himself. He's clearly losing himself. I love these paintings because it can tell us how it must feel to have Alzheimer's disease. We all know what happens, but this is how it feels. It's a loss of self. It's progressive, and it's the sixth leading cause of death. And of course, what we want to do is eliminate early as possible the neurotoxins that cause the disease. And we are getting closer. So you probably know very well that the FDA approved the first new medication in 20 years back in early 2021, an antibody called aducanumab, priced at $56,000 a year. It's had a lot of uncertainty and is now no longer on the market. But I would say we're getting close. What's been going on is to try to find the right toxins to target. And we know since 1906 that there are plaques and tangles that show up pathologically in someone like Augusta D., the first Alzheimer patient. And we still diagnose Alzheimer's disease as dementia with plaques and tangles, but I think that's a bit off the mark now. So lots and lots of different approaches, pharmacologically, cell biologically, and especially human genetics have led the field to target amyloid plaques for therapeutics. And there have been vaccines that have developed, small organic molecules and therapeutic monoclonal antibodies, like I just mentioned, the aducanumab. Regrettably, these have amounted to a huge, costly failure. And what we can think about, from my perspective at least, is that the amyloid plaques are the wrong target. What you're looking at here, by the way, is an atomic force microscope image of an artificial amyloid fibril. starting with synthetic A beta put in a test tube, allowed to incubate with itself for 24 hours, put on a mica plate, and then imaged by AFM. So you're looking at individual fibril molecules, which look very much like what you see in an amyloid plaque. This is the wrong target. The right target, from our perspective, and I'll try to convince you this, is a much smaller form of an A beta assembly. So we take, in this experiment, the same A beta that was synthesized by a chemist and put it in a test tube and adjust the milieu. And instead of getting fibrils with the same starting material, we end up with fibril free preparations of A beta oligomers, again, seen by AFM. So these are 2 nanometers in height. Each one of these is an individual molecule. And this was some discovery work begun with my good friends and colleagues, Grant Kraft and Caleb Finch, who got excited when we discovered this and the following couple of data points, and started a company called Acumen Pharmaceuticals, which targets A beta oligomers for therapeutics. And I'll mention them later. So, what we found was not only an interesting structural biology issue, but the fact that those fibril free preparations were potent CNS neurotoxins. They impaired LTP, as you see in this hippocampal tissue slice from a mouse. LTP normally kicks in right after a burst of activity, but in the presence of A beta oligomers, which we first called ADDLs, to distinguish them from amyloid. Uh, and we see that that potentiation does not last. It's not long term, it's inhibited. On top of that, if we continue to expose these tissue slices to A beta oligomers, what we see is significant cell death. Without and with ADDLs or oligomers, we see a great amount of cell death due to the exposure, unless we use tissue from a fin knockout mouse. And here we see an absolute resilience in the face of the A beta oligomers. And that led us to say this is not molecular shrapnel like people thought amyloid plaques were in the old days, but rather part of a dysfunctional signaling process that was triggered and instigated by A beta oligomers. These types of experiments led us to introduce, as Maira pointed out before, we introduced the A beta oligomer hypothesis. We said that the damage in neurons leading to Alzheimer's disease is due to the impact of these toxic A beta oligomers. Since that time, there have been over 5,000 papers dealing with A beta oligomers and their relationship to Alzheimer's disease. And we now can link these as upstream molecules that impact many aspects of Alzheimer's pathology, aberrant tau phosphorylation, plasticity dysfunction, as you just saw, and so on around the clock. And let me just show you a couple of examples. A beta oligomers, if we take some very mature hippocampal neurons and put them embryonic and let them mature in cell culture, say for 3 weeks, they develop axons, they develop dendrites, they develop synapses. And if we expose those cultures to A beta oligomers, this is what happens. This is a bit of a dendrite. We're looking at dendritic spines stained with an antibody against drebrin. After 3 hours exposure, we see that these spines on the dendrite have changed their morphology. So they have deteriorated into this very immature looking spine. And as a matter of fact, if we continue to expose it, these even immature looking spines disappear. Are these relevant to function? I would argue for sure. These are images from human brain samples looking at a dendrite with its spines versus someone who has Fragile X syndrome, mentally retarded, and we see that the spines are totally degenerated with respect to normal, and amazingly, they show a tremendous similarity to what you see with A beta oligomers. That was an in vitro experiment for us. Here's some work from Fernando Di Felici and colleagues, in which the A beta oligomers have been injected into non human primates. And what we see here is a sham injection stained for phosphorylated tau at tau phosphoserine 396. After injections into cerebral ventricles with A beta oligomers, we see a marked upregulation of this Alzheimer's form of phospho-tau. Another in vivo experiment, which is even more compelling, is the injection of wild type mice. And what we're looking at here is some rather outstanding data by someone named Dr. Mayura Pika, where the animals were being tested for their ability to learn a novel object recognition task or a novel placement location recognition task. One is associated with the hippocampus, one more cortical. If we inject these animals with vehicle and test 24 hours later, we find that, as you see in the orange bar, which is for the new object or the new location, uh, the animals typically love the novelty. However, if they've been injected with A beta oligomers, they don't remember what's old and what's new. They can't tell the difference. Similarly, with the cortical tasks, vehicle injection, love the new, A beta oligomer injection, can't tell the difference between old and new. So when we started the field, this was the concept that the damaged neuron that leads to Alzheimer's disease was due to interaction with amyloid plaques. Amyloid plaques made from amyloid beta peptide, A beta 42, they form fibrils which associate in plaques. After all of those types of experiments were done showing what A beta oligomers can do in the absence of fibrils, we now see this image from the NIH progress report. Sorry, I'm getting a sign that's interrupting me here. And we see that the damaged neuron can now be accounted for by the A beta oligomers. In fact, the director of neuroscience at NIA, Eliezer Mazda, who also has worked in this field, wrote in a review, progressive accumulation of A beta oligomers has been identified as one of the central toxic events in Alzheimer's disease. And of course, what we've been looking at so far is experimental models. What we wanted to do is establish the human disease relevance, and the key advance that made this possible was to develop A beta oligomer selective antibodies, antibodies that were not going to bind to monomeric A beta, of which there is a lot, not going to bind to amyloid plaques, of which there is a lot, but it's just going to bind to the small soluble oligomers. And these antibodies were crucial in allowing us to show that A beta oligomers, in fact, are central to Alzheimer's pathology. What you're looking at here is a series of dot immunoblots up at the top with samples taken from patients who died with Alzheimer's disease. You take a high-speed spin 100,000 G supernatant to make sure there's no fibrils present, dot that onto the filter paper, stain with our antibody, and compare that with what you see in control. And what we see is that, in fact, the human brain soluble extracts are showing an Alzheimer's dependent signal in these immunodot-blots. These originally were done with polyclonal rabbit antibodies, which were extremely good at making antibodies that were selective and potent. If we look more closely at the structure of that molecule that's giving us the signal, we do two-dimensional Western blots. So we take these extracts, separate them first by isoelectric point, and then by mass, then do a blot and stain with the antibody. And what we see is this little grouping of molecules that are 54 kilodalton with an isoelectric point of 5.6 from an Alzheimer's brain extract that are absent from a control brain extract, but are present in the types of preparations that we use for experiments. So 2D Western blots show these 12 MERS, 54 kilodaltons, both for Alzheimer's extract and for synthetic oligomers. Now, a lot of work, of course, is done with transgenic mice. A particularly robust form has 5 different transgenes in it, developed by Bob Vassar, who discovered BASE and is also on the faculty here at Northwestern. And we use his mice because they develop problems early, so we don't have to wait too long for experiments. Here we're looking at oligomer appearance as the mouse ages from 2 months to 3 months to 12 months. And at 2 months, we see essentially no A beta oligomer signal, but this begins to show up between the end of 2 months and the beginning of 3 months. And we see the presence of oligomers in the subiculum, which then starts to spread out into the CA1 region and ultimately into the dentate gyrus as well, and is quite robust by the time the animals are 12 months of age. What's important to us, of course, is that the onset of memory dysfunction follows that buildup of A beta oligomers. Here again, we're looking at Maira's novel exploration time tasks, hippocampal dependent in this case, looking at 2-month and 3-month animals. The transgenic mice really are as good as the wild type at this early age before the oligomers show up. But once the oligomers are there, we see that the transgenic animals start to lose this capacity, and by the time 6 months rolls around where the oligomer buildup is robust, we see complete elimination of memory in memory formation in these transgenic mice. Age-matched controls are happily performing. So we see a dysfunction, we see pathology that's induced, we see memory loss, we see it present in humans. What we, of course, want to pursue next as scientists is to go after the mechanisms that underlie this. And I mentioned at the outset that Finn was a early signaling molecule that was first linked to the function of A beta oligomers. That's been picked up more recently by Steve Strittmatter at Yale, who's done a great deal of interesting work with it and related pathways. This is from a review from Distruper and colleagues indicating that there's a real spider web of different interactions that are instigated by this binding of A beta oligomers. And it's that question that we're spent quite some time about. How does that begin? So those are all downstream events, intracellular, but A beta oligomers I think are extracellular when they're added exogenously. How are neurons attacked? So here we're looking at hippocampal neurons that are differentiated in a dish. They have synapses that are formed. They're from rat embryos cultured for 3 weeks. And what we do here is to increase the dose of A beta oligomers, and we see a saturation curve. And we see a high affinity, 60 nanomolar or so in total A beta in oligomers. So at this stage, we're not quite certain what the stoichiometry in terms of monomers is. If we assume that there's 10 monomers, let's say just for convenience, per oligomer, then the midpoint for this saturation would be 6 nanomolar, sorry. We also see this type of binding in synaptosomes. So it's saturable, and it's cell-specific. Here we have 2 hippocampal neurons stained in red for CAM kinase 2 and stained in green with our oligomer selective antibody. And what you see is that it's primarily the neuron on the right and not the neuron on the left that has all of these oligomers bound to it. The co-localization is shown in gold. The neuron on the left largely has no binding oligomer to it. What are those hotspots? One can imagine lots of different domains. It's clear, though, that they co-localize with a postsynaptic marker that's essentially 98% specific for synapses in hippocampal nerve cell cultures. This is postsynaptic density 95, antibody against that. If we double label with PSD95 antibody and oligomer antibody emerge, what we find that is over 90% of the oligomers are binding at the post synaptic density sites. And looking at it more closely, we can in fact see that it is at the postsynaptic site as well. Here we're looking at a beautiful hippocampal neuron that's been matured in a dish, stained with CAM kinase 2 in red, stained for oligomer binding green. You don't see any green because it's all co-localized on this neuron. And we see that it's the synaptic spines where the majority of the oligomers end up. This binding is antagonist sensitive. So in an early stage screening of a chemical library for blockers, we found one that is effective in preventing the binding of oligomers. Here's control binding. We see the binding to the spines along the dendrite of the oligomers, 100 nanomolar A beta O, 10 nanometer, 10 nanomolar A beta oligomers. In the presence of 1 micromolar ATA, this is blocked. So this requires available sites to bind to it. ATA would be great if it were specific, but we can't use it for therapeutics. But because it's a very promiscuous type of molecule. It binds to lots of different targets, so it would not be of use for therapeutics. But it does show that the binding is antagonist sensitive. And furthermore, that binding site is a protein. So if we trypsinize either cells or solubilize synaptic membranes, in the presence of trypsin, binding is reduced more than 90%. So these oligomers bind to proteins. The take-home lesson from all of this is that A beta oligomers act as pathogenic ligands for cell surface toxin receptors. And the same is pretty amazing to me. The same result can be obtained not just with synthetic oligomers, but with Alzheimer's disease brain extracts, soluble extracts from AD brain, given to hippocampal cells, bind in this same pattern of attachment to synapses as synthetic oligomers do. The control brain extracts don't bind at all. When I saw these results, this was the first time I really believed that something for sure was going on, that this was, in fact, part of the disease process, not just something that shows up in a test tube. Well, what are those receptors? There's still an international hunt for those. There's a menu of different receptors that goes on and on. Prion protein from Steve Strittmatter has caught a lot of attention. mGluR5, which we found, and Steve is actually linked to the mGluR5. There's an interesting protein from Carla Schatz in the retina that I'll talk about in a bit if I remember. The question is, what are those? And do they play a role, and how do they play a role? This is an interesting experiment in which we used a new approach to knockdown called spherical nucleic acids, which was invented in the laboratory of Chad Merkin, who's using this to deliver antisense to neurons and other, actually for cancer therapy as well. In this case, we have two different cultures that are labeled with the oligomers or with the synaptic marker, drebrin. In the absence of antisense with these SNOs, what we see is lots of oligomer binding and a great loss of synapses. If we come in with mGluR5, however, and knock it down with antisense, we see very little binding and retention of the synapses. So that was pretty exciting, which pushed us to go one step further. And we worked with Antoine Triller and postdoc Dr. Renner in his lab. Pascal Lacour from our lab spent some time in Paris doing these experiments where you can take A beta oligomers and look at them as individual molecules on the cell surface of cells by tagging them with a quantum dot. And what we see, very interestingly, is that the A beta oligomer bound to the cell surface moves around almost in a random walk to begin with, but after 5 minutes, it stops. And this is at a site of a synapse. If we do that, if we wait 60 minutes before we track the individual motion, we see it doesn't move at all. So it's become immobilized at synapses with time. That reveals to us these quantum dot individual molecules in living neurons show the presence of some type of synapse trap. And a partner in crime here seems to be the hijacking of these mGluR5 receptors. Because if we look at the mGluR5 receptors with a quantum dot, we see that they move all around to begin with, and then after 5 minutes, stop moving. Or if we start these experiments after 60 minutes of exposure, they don't move at all. So there's been a trapping of oligomers at a synapse and a hijacking of mGluR5 to go along with that. Our evidence suggested that the mGluR5 per se was clearly involved, but not a binding protein of high affinity on its own. We worked closely with a group at the University of Illinois in Champaign-Urbana, headed by Stephen Sliger, who invented a beautiful technology called the nanodisk, which in essence is a nanoscale artificial membrane. So what he does is to take some lipid and some membrane scaffolding protein. One protein takes a preparation that's exposed to detergent, in our case, the synaptic proteome, and then cause the self-assembly by removing the detergent. And what it assembles is individual nanoscale membranes, each with a different protein in it. So we have solubilized membrane proteins, but still in a membrane environment. And that allowed us to do a immunopurification of proteins that bound in this in vitro state to the A beta oligomers. And what we found was that the sodium potassium ATPase alpha 3 was a very good binder of the A beta oligomers. Alpha 3 is interesting because it's involved in recovery from long term potentiation. It's a neuron specific form of the sodium potassium ATPase. There are multiple ATPases, of course. What we're doing here is again look at a hippocampal nerve cell culture and studying the distribution with an antibody of the alpha 3 subunit, and we see that it's speckled across the dendrites, which are localized to synapses. This is relatively shortly after A beta oligomer addition, but after 60 minutes, we see a significant redistribution of the ATPase molecules into big clusters. And those clusters also contain amyloid beta oligomers. This is very interesting because independently, Minako Hoshi's group in Japan had identified the alpha 3 as a target for amyloid beta assemblies isolated from Alzheimer's patients. And was able to link the alpha 3 to the death of neurons induced by the amyloid beta oligomers. Another interesting facet of this to me is the fact that ATPase is not just an enzyme for moving sodium and potassium, but it also serves to organize protein protein complexes within the membrane. And what we see, of course, here is that membrane distribution of ATPase is changing. So the docking stations are changing. That led us to this hypothesis that we have a alpha 3 subunit that interacts with different proteins in the membrane to cause a cluster. But if we then add A beta oligomers, which interact with other alpha 3 subunits, which then come in and create a different domain, which you might call a toxicity domain, that leads to a great increase of calcium leading to toxicity. That may not be right. The great South American author Borges, in his detective stories, said, "Your hypothesis doesn't have to be right, it just has to be interesting." And I still find this a very interesting idea, which may be right. So we have a lot about the pathological presence of the oligomers and their action as a toxin and how those toxins work. We need to ask ourselves, are these of translational value? And let me start by talking about targeting oligomers for diagnostics. Um, I mentioned Chad Merkin before, who is a nanotechnologist, inventor par excellence. I told you about his new way to develop, to introduce antisense. This was an ultra-sensitive single molecule assay that he developed quite some time ago called the biobarcode. And we worked with him to ask the question whether or not A beta O's might be a marker in the CSF for individuals with Alzheimer's disease. And in fact, this is a super sensitive assay. This is femtomolar levels here, and we're taking femtomolar amounts of A beta oligomers and in a 10 microliter volume, and looking at control populations, which are in the neighborhood of femtomolar. But if we take the CSF from individuals who had been diagnosed with Alzheimer's disease, we see in this semi-log plot a marked increase in the oligomer levels, 30-fold for the median here, with almost no overlap between the oligomer levels in Alzheimer's CSF versus control CSF. We're working with one of our colleagues from these days, Jeff Anker, now at Clemson, to develop a simpler way of doing this that is now in progress. So I think we might be able to find oligomers in an AD-dependent manner in the CSF and perhaps even in blood. So that was for clinical chemistry, but we can also consider the possibility of using it for brain imaging. In fact, what we have done is to take our antibodies that are selective for A beta oligomers and try to do molecular MRI. And in this case, what we do is take the antibody, put a nanostructure on its end, which is a very good beacon for the MRI instrumentation to pick up. And what we see here is with one antibody, we see clustering of A beta signal in the hippocampus of the 5X mouse, but not in the wild type. And this is a different antibody down here, again, looking in the hippocampus, so wild type down here, clean in the hippocampus versus 5XFAD hippocampus, speckled with signal. This is with a higher magnification or higher magnetic source here. And quantitatively, if we integrate the transgenic versus the wild type, we see a marked increase. It's even more vivid if we make a PET probe instead of an MRI probe. So now instead of putting a nanostructure on the end of the antibody, we put a copper 64 chelator. These are the controls. These are 9 brains from 5X mice given a control IgG with the copper. These are wild type mice given our PET probe, wild type mice given an IgG with the copper 64, all of them at the bottom end of the heat map. But if we take the 5X FAD and give it our PET probe, we see the region of the brain showing a incredibly bright signal relative to the controls. So this is a kind of, for us, an exciting alternative to looking at amyloid plaques, as is the current common practice. So I'd like to move from diagnostics to therapeutics and talk a bit about the next generation of Alzheimer's immunotherapy. And since we got an early start on this, we've been targeting A beta oligomers because of our experience in this mechanistic approach. So we're trying to develop immunotherapy that targets oligomers and not plaques. There are two antibodies that are currently of interest. Biogen's aducanumab was the one that I first mentioned with the New York Times article. It has a bit of efficacy, but it requires high doses. So it actually can interact with oligomers, but it's not selective. It binds every bit, if not better, to amyloid plaques, which of course means that a lot of the antibody gets sponged off because there's a lot of surface on the amyloid plaques, which is why we would guess that such high doses have to be added. Now, the company that Tuck Finch and Grant Kraft and I started, Acumen, developed an antibody in partnership with Merck over a 7 or 8 year partnership that doesn't bind to amyloid fibrils, doesn't bind to monomers like Eli Lilly's antibody did, their early one, but in fact binds to, in a very selective way, to the A beta oligomers. Now, this antibody is called ACU193, and it's highly selective for oligomers in AD brain. These are patient specimens, aged brain, no binding, intermediate brain, and I think this is quite an old person, around 100 years old, only intermediate pathology, showing the oligomer buildup and advanced Alzheimer's, showing a great deal. These ACU193 antibodies do not bind to dense core plaques, but they will bind to the halos of oligomers that are found around plaques. This is a ELISA type assay for the same. These A beta oligomers that are stained with ACU193 are cell selective. What we're looking at is the ACU193 pattern on the right here. This is ACU193 in yellow. This double labeled for this phosphotal is labeled with AT8. It's a serine 202, 505, 205. And this is unique to Alzheimer's disease. We see that wherever there's an ACU193, there also, no, the other way around. Wherever there's an AT8, there also is an ACU193. But in some cases, we see oligomers present in neurons here, and here, for example, that don't have AT8. So I would infer from this that these neurons are in a slightly different stage of their cascade of pathology than the neurons where the AT8 is present. These antibodies, in fact, that stain so selectively are very good at rescuing memory in the 5X mouse. So here we look at wild type, time spent on the new versus time spent on the old. 5X FAD can't tell the difference unless they're given a beta oligomer monoclonal antibody. In this case, ACU193. In this case, NU4, another monoclonal antibody of that same family, in fact. When MIÉRCOLES got these data, and in fact, she said, if I ever start to have memory problems, please give me a shot of this stuff. And in fact, there are people out there now who are getting this ACU-193. Acumen started a clinical trial last October. It's called Intercept AD. The phase one results will come in this coming winter. And this is not just healthy adults, and in fact, it includes participants who have mild cognitive impairment or mild dementia due to Alzheimer's disease. So, in addition to pathology and safety concerns, these results that will come out in the winter will give us an assessment of the efficacy of ACU-193 in rescuing memory and the ability to perform daily activities. So this is pretty exciting, and certainly, like everybody, would keep their fingers crossed. So we've talked about this toxin. Why preserve nature? Why preserve evolution? A toxin forming A beta 42 sequence. This really likes to form toxins. You can take a 10 nanomolar dose, put it in a test tube, and it forms oligomers, and they're toxic. Why have we preserved that? Well, it turns out that we've just discovered and published this last year that they have a physiological role. A beta O's have a role in neurodevelopment. And what we're looking at here is the retina, which we finally are going to be talking about in the last couple of minutes. This is a chick retina, which hatches after 3 weeks. This is at the end of 1 week. And what we're doing is orienting the retina so that the light comes from the top, goes through the retina, and interacts with the photoreceptors down at the bottom. This is an antibody called NU4 that has been given to these retina tissue, and you can see after 1 week in development that the oligomers are beautifully staining the nascent optic nerve, the nerve fiber layer. That's after 1 week. This is where it is after 2 weeks. It's moved on from the nerve fiber layer into the ganglion cell layer. It's found in the synaptic layers in the inner plexiform layer. It's in part of the internuclear layer in the amacrine cell layer. It's found in the horizontal cells and down into the pigmented epithelium. So we've had this spread in a cell-selective manner between week 1 and week 2. And at week 3, it disappears. So all of these oligomers that were present so robustly when so much differentiation was going on have gone away. And the chick, why study the chick? It's very easy to do developmental studies in the chick because they're so nicely packaged for us. But more than that, the avian retina has an A beta sequence that is identical to the human sequence. So the A beta 42 of chick is the same as the A beta 42 of humans. That means the potential exists for making oligomers, and in fact, for a brief period in time, they are indeed present. That brings to mind this New Scientist article that showed up a year ago referring to prion proteins that might have a distant cousin here with the oligomers, the Dr. Jekyll and Mr. Hyde proteins from a Robert Louis Stevenson novel. The good side, Dr. Jekyll; the bad side, Mr. Hyde. Mass spec confirms that these A beta oligomers are present by looking at an isolated fraction that contains the oligomers and a separation by gel-free electrophoresis. Running through the mass spec, we get the same sequence for monomeric A beta as humans in this 45 kilodalton band. So this is roughly a 10-mer that is made up of A beta 42 monomers equal to each other in terms of their human sequence. This is a series of experiments that are shown in Bartley et al. In 2022 in the International Journal of molecular science. This mass spec was also done by Mayra and a couple of her colleagues from Brazil up here. The Western blots, in fact, if we go further, show three different proteoforms. We were just looking at this proteoform here, but there are other bands that show up with the other antibodies that we use routinely. NU2 is an antibody that stains roughly a 10-mer. ACU193 that we were talking about looks at different bands in this preparation, physiological, and NU4 looks yet again at different bands. So we have three different proteoforms. Now, I showed you the NU4 proteoform, which I didn't mention, but the first one was the NU4, and it downregulated. Well, the NU2 distribution also shows a developmental staging program that downregulates, although the distribution, as we'll see, is slightly different. But it's very interesting. This NU2 proteoform co-localizes with this same form of phospho-tau that we've been talking about, AT8. So this is an AT8 stain in which there is the amacrine cell layer that's labeled here. First, it appears in the ganglion cell layer, just like NU2, just like NU2, and just like NU2, it disappears. So the phospho-tau that's stained by AT8 disappears at the same time that NU2 does, suggesting, doesn't prove by far, but suggests the possibility that these oligomers are involved in a signaling pathway that involves the phosphorylation of tau. Now, A beta comes from amyloid precursor protein, which also, of course, is present in the retina, also has this developmental pattern, but somewhat surprisingly does not downregulate fully just before hatch. It's still present in these layers, unlike the NU2 and NU4. However, the ACU193 proteoform is still there. So we have a third pattern of development distinguishing these three. So we have three proteoforms: the NU4, the NU2, and the ACU193. At E14, they distribute similarly but differently. Even in the interplexiform layer, we see the NU4 bands are distinct from the NU2 bands. And then just before hatch, NU4 forms, NU2 forms disappear, ACU193 is still around. That strikes me as an incredibly interesting pattern for trying to understand developmental neurobiology. This proteoforms of A beta oligomers have to be under some type of spatial-temporal control that is complex and interesting, and as we'll see, significant. If we look at a close-up of those A beta oligomer distributions around a neuron, confocal shows a bit of a blur in the cytoplasm, but if we look at images with a super resolution scope, where this is 1 micron, we see that the A beta oligomers are packaged into vesicles. We don't have an identification of these yet. They are some sort of trafficking vesicles, I assume, and it's quite likely to me that these are involved either in the synthesis of the A beta oligomers or the trafficking and release of oligomers in extracellular vesicles under study. To get at the possible function of oligomers that are released, we developed, for our use, a ex ovo embryo with some help from a colleague, Bruce Dunn, up in Milwaukee. And what you're looking at here is what a chick looks like if it develops in ovo for 14 days, or what a chick looks like in our hands. This is from Hamburger and Hamilton, classic book. In our hands, if it develops without a shell. So without a shell means that we have access for injections of different antibodies or drugs. And the eye is particularly accessible for these, and we use that to microinject, in the first case, an inhibitor of the enzyme required to make A beta. So this base inhibitor causes retina dysplasia. So what we're looking at here is a slice of the retina This is the back of the retina here. And if we look closely at these little arrows, what we see is again, the light is coming from the top to the bottom, from the top to, oops, sorry, the top to the bottom. This is stained with DAPI, which stains nuclei. And we see that the back of the retina is dark, but being disrupted are these rosettes that are dysplasic that come off of the back of the retina, seen in lower and higher magnification. If we look at non-injected eyes or phosphate buffered saline injected eyes, these structures, these rosettes, do not show up. So this is what happens if we eliminate all production of A beta. This is what happens if we target A beta oligomers, whether with our NU2 antibody or the ACU193 antibody. The consequence is, just like the base inhibition, is a disruption of cell placement in development. At 2 weeks, the retina should be nice and layered and flat. This isn't what happens. Instead, we have these rosettes coming off from the back of the retina. So I would just end by saying this leads us to a new hypothesis, and that is that A beta oligomers, which we and so many others have linked previously to Alzheimer's pathogenesis, and there's 5,000 papers on that link. We would say that they also appear very briefly during neurodevelopment to help in histogenesis and connectome differentiation. And this is something of keen interest to us. And I think if we can learn more about that function, and even more importantly, about that regulation, I think it will give us new insights into what's going in Alzheimer's pathology and pathogenesis. So let me finish just by saying we've had terrific teams over Northwestern in the last 20 years. This is one team that was particularly outstanding. And I'd like to call attention to that. We've been supported by a number of associations that I appreciate a great deal. And I also want to give thanks to my good friends and collaborators who've been very important for this work from Brazil. Fernando and Maria Cristina de Mello, Sergio Ferreira, Fernando Di Felici, Adriano Cebulella, and Mayura Bika. So with that, I'd like to thank you all for being with me, and I'm going to wrap it up.
Thank you so very much, Bill. As per usual, you don't disappoint. Like, your talks are very enlightening and very easy to follow. You are very. I said that before so many times. You are very good with words. Like, you talk in a way that always mesmerizes me, and I can't cease to learn with you. Thank you for.
I'll tell you, Mayura, that's a sort of question I love to get.
So we were very delighted by your presentation. I believe that everyone in the audience was like very interested in what you had to say, and in like showing the potential role of amyloid beta oligomers in physiology. I think that's a very interesting thing, and then we need to keep looking into it, because there is a reason why these oligomers are in there later. It's like, it seems it's because they are needed at the initial stages, so they have a physiological role. And then maybe if we understand better the physiological role of the oligomers, then we can tackle it better when it turns out to be a disease.
Can I mention just a bit on that? In fact, makes a lot of sense what you're saying. So one can imagine other developmental programs that get re-expressed when injury takes place. Now, even a simple system like the neuromuscular junction tries to go back to a younger age and recreate itself. Mm-hmm. So I would say that you and I are on the same wavelength there.
Yeah, amazing. So amazing, Bill. I have one question for you. It's just like a kind of like a biased question, because I'm so into the work that you presented today. So before I do my question, I'm gonna actually open the floor for the audience because I'm pretty sure that they might have questions for you. And then in case that they don't have any questions, then I'm gonna shoot my question. So
Sure.
right now I invite the audience to ask Bill whatever you want about the work that he just showed. You are welcome to use the chat. I can reproduce your questions. For the young students, like, don't be shy. If you want to ask in Portuguese, it's fine. We can translate it. So just so we know that we are open to questions.
And then just in case somebody has this question, I can say that no, in January and February, it never gets cold in Chicago.
Let's see. Usually the audience is a little bit shy, young students.
Absolutely.
Yeah, so I'm gonna go ahead and ask you. So I'm gonna start. Maybe people get a little bit warm up. I was like, I think it's something that we discussed in the past a little bit, and then I would like for you to say a little bit more about it because since I left the lab, you guys made a lot of progress, and then I'm very delighted to see the data that you showed. Do you think that might be a link because so many articles have talked and like described the link in between eye disease and Alzheimer's, like for instance, glaucoma, cataract, like so, and then as potentially showing those as also risk factors for Alzheimer's disease. So, like you just showed us how important oligomers are to the eyes at the beginning. So, do you think there is a link afterwards, like in between these diseases and Alzheimer's as well? So, like, do you think that that's the data that you generated is going to help us to understand these potential risk factors for eye disease and Alzheimer's?
I think that's a fantastic question and a real possibility. We actually have started to look for oligomers in humans with AD in their eye. So we're working with Maggie Flanagan, who's a neuropathologist in medical school, who just took the ACU193 antibody, in fact, and in initial studies showed that individuals with Alzheimer's disease had elevated signal due to the ACU193. So I think it's showing up. There's maybe one paper or so that suggests that this may happen in transgenic mice also, and something that is perhaps very early in the disease. They thought the possibility was that it comes through the blood to get into the eye and the retina, but their speculation didn't have the information that the retina is capable of making oligomers on its own. So I think that whatever causes well, we can go back to some work we did with Peter Fredrickson a decade ago, where we were studying a different model. And this was the wild type rabbit, which, like a chick, has the same sequence as human A beta. And Peter and his lab made the rabbits type 1 diabetic using streptozotocin. And what we then looked at was the fact that type 1 diabetes caused the buildup of oligomers in the rabbit wild type brain, including in the retina. So that was pretty interesting. And I think we're almost circling back to that right now with the chick instead of the rabbit. They're less furry.
Yeah. Very nice, Bill. Thank you so very much. Since the audience is a little bit shy, and we are close to end the session, so we have only a minute left. I thank you so much for accepting our invitation one more time. It's like it's such a happy moment when I see like people that I admire and that inspired me so much to present data that it's just like fantastic. And as you said, you have like a good long collaboration with people from Brazil. We hope to get you in person the next time so we can have like a really great time together. Thank you everyone for joining this session. Thank you for watching and participating somehow. Before we go for good, I would like to say I would like to invite you all. Like I've been doing this in all of the sessions that I'm chair. I would like to invite you to go to the exhibiting hall because we have like really nice stuff being showed there, like videos honoring Professor Mauricio Rocha Silva and Professor Sérgio Ferreira, which are like really great pharmacologists. So like young pharmacologists that just joined the society, that just joined the conference. They want to know more about the history of pharmacology in Brazil. Should go there and then check it out. Also, please go to the Young Committee booth. So we have a stand for this BEFT Jovem. So that's the young committee for the society. We have a raffle going on for a book, so you can participate on that if you visit this stand. So like, please go. I'm emphasizing how important it is, even though we are online and understand that everyone is a little bit tired of it. That to be interactive and then to socialize as much as you can. So thank you so much, everyone. Thank you, Bill. Thank you for being here with us. And I end up this session. Thank you.